Okay, so you all are doing, wanted to do research in the area of different field. Okay, and in case of research, there is you should have a tool to investigate something. Okay, and there is and what this tool is for? Tool is for to demonstrate some kind of what you said, some kind of evidence, to produce some kind of evidence that what you observe, yeah, definitely you learn, you come to know about that observation, or you saw that observation, you make your own opinion that this is the thing which I am looking at. But you need to tell to everyone. For that, you need evidence which can be reported. Okay, so, and one of the important tools for doing research is microscope. Okay, microscopy is needed because microbiologist knows that there are tiny living organisms which you cannot see from your naked eye. So and for that you need to use some special kind of instrument which will able to see that organism. So that's why there is a microscopy, means the micro thing you can watch. So that is the meaning of microscope. You might be heard about the telescope. You might be heard about telescope. So telescope means the thing which you can visualize the things are very far. So, and here the things are near to you, but you cannot observe from your naked eyes. So that's what this microscope. Microscopy is a very important tool to produce direct evidence in research. Direct evidence in the research means it's like, sometime when you go to somewhere, let's say you go to Gujarat or anywhere, or you are here at the class. How someone can know, or your father or your parents come to know that you are in the class? So then, what evidence you are going to produce then? If they really doubt on you that that you are not in the class, then what you will do? You will take picture that you are sitting in the classroom with your friends and with the teacher. So what you did, you capture an image to sort their doubts. Because in science there is always everyone is doubting on everyone. When you doubt, when you ask the problem, only then science will progress. ऐसा नहीं कि उसने ये कह दिया ठीक ही होगा. Nothing like that. There should be some evidence. On the basis of that, you can report your finding. And these microscopes play a very important role in produce that kind of image capture. Okay, so I hope that now you understand about the microscope. That what is the microscope? Microscope is the thing where you can look for the tiny objects such as bacteria or any kind of stain or anything which is not visible from the naked eyes. So that is microscope. Now today we do have to cover few things in microscopy. Just what topic we are going to cover. So scanning probe microscopy, super resolution microscopy, fluorescence microscopy, which includes film and fragment. So I will send these three. So next I do have the 4 classes I have to take, and in next 4 classes these 3 microscopy, scanning probe, super resolution and fluorescence. So today I will cover the fluorescence microscope. Okay, so you know there is a microscopy. Microscopy is a kind of microscope is an instrument, and microscopy is kind of discipline where you analyze, where you take those pictures. So that's what it is ending with the microscope. Okay, so then now coming to the let me show you the first slide. So today we are going to cover the fluorescence microscope. First, we need to. You are very familiar with this kind of picture. You might be observing in different labs or histopathological lab that this is commonly called as a microscope. So it's a simple microscope where you can observe any tiny material which is not visible from your naked eye. Okay, this microscope, when you really want to see, there should be a contrast. Contrast means when you want to clearly see something. Like sometime when you saw that, have you heard the one terminology camouflage? Camouflage. You know that? So in that, the animal is change their color according to the environment, so that you cannot observe. And you cannot kill them because there is no contrast. So for microscope, to better visualization, we need to stain them. We need to give the color to them. Okay, so if we can give the color, we can easily observe. जैसे आप लोग पार्टी में जाते हैं, तो पार्टी में ऐसा नहीं कि कोई भी डल कलर पहन के चलेंगे. वहाँ काफी मतलब अगर भाई let's say you go for some function, you always wear dark color thing. हो सकता है लाइट कलर ही पहनते हो, पर अभी इसको समझने के लिए ठीक है. So that's why this microscope needs some staining. That's why this is known as a bright field microscope. Bright field means there is an illumination of light, very bright light, and then this bright light will go and it will difference between it will give the better visualization of your stain. So this is the gram staining of the bacteria which you can observe. So this is taken from some microscope. This picture is taken from the bright field microscope. So again, we are understanding all these things, whatever the weather, it's a bright field microscopy. Next, we are going to understand phase contrast microscopy. And then third, definitely the one which we are going to know about is a fluorescent microscope. Anyone knows about the fluorescent microscopy? आप में से किसी ने कभी fluorescent microscopy के बारे में नहीं सुना ना? Nothing.
That means fluorescent microscopy, the process is possible because of movement of the electrons. When the electrons move from ground state to shattered state, after 10 power at -8 seconds back into the ground state, that means that it releases some color for the fluorescence.
So there's during our maybe intermediate classes, we read about the fluorescence. That what is the fluorescence? So we will, in this class, we will learn about this, but it's good that only you know. Majority of the students are not knowing, so that it's good that I am teaching. Otherwise, if already student knows, then what is the point of teaching? So, but whatever you said, that fluorescence phenomena that you told. So exactly on this phenomena, this fluorescence microscopy works. So it's right. But here we are looking at how this fluorescence microscopy help in the research point of prospect. But whatever you said, it's right. Let's see. So bright field microscopy. So you understand here in this microscope there is a kind of bright field. This is kind of a bright light is passing on, scattering on this your specimen and giving you a picture, visualization. So this is the gram staining picture. Now second is phase contrast microscope. Anyone knows phase contrast? What is this phase contrast microscopy?
Clear image.
Clear image. Okay.
DP से बताता है कि जो चीज जो हम नहीं देख सकते हैं, it should be done by the phase contrast. So contrast color.
Okay. Anyone else? So whatever he said is right, that phase contrast is this microscope. So again, microscope is this. Now we are understanding the term phase and contrast. Okay, so because, and one thing I will tell you, that these phase contrast microscopy is used to see the live cells. Okay, any living cells, if you want to observe, you need to use this phase contrast microscope. Like, let's say if you are in a cell culture or cell culture room, you definitely, there should be a phase contrast microscope. Now, the first terminology, phase contrast, because there is in cell, there is a different refractive index. The cytoplasm has a different refractive index, and outer area, which is the media, has a different refractive index. So that's why, being different refractive index, or you can say density of the solution in cell, cytoplasm is little bit more denser in comparison to the outside. So it's produce a gradient. So this microscope will differentiate on the basis of this and this. So it will give you image. That is on the basis of, so it will change the phase. So that's why, and phase on the basis of refractive index. So that's why it is called as a phase contrast microscopy, and this is happening due to the different refractive index of the media that is contained either in the cytoplasm or into the here. So generally, live cell imaging, or if you want to see any cell, you use this microscope. Every day, you cannot use fluorescent microscope. अर्धे ऐसा नहीं कि fluorescent microscope से ही देख रहे नहीं. Phase contrast microscope. Now the third is fluorescent microscope. So fluorescent microscopy. Now the fluorescent microscopy. Why we need fluorescent microscopy? This is the one question. The answer is that inside the living being, everything is a no color. कोई कलर नहीं. हमारी जिंदगी में बहुत कलर है, बट इनसाइड द लिविंग आप देखिए कहीं कोई कलर नहीं. देयर इज नो कलर. ओके, सो व्हाट यू नीड टू डू टू गेट बेटर विजुअलाइजेशन, यू नीड टू ऐड कलर. ओके, आप जैसे लेट्स से दिस इज द वॉल, so nothing is there, but you add some color. So if you add some color, it will be immediately you can observe that, or it will look more beautiful because everything is in a yellow paint. But if you want to draw some cartoon or any image you want to hear, you will put some color. So same way, here in the case of the fluorescent microscopy, you add a molecule which is known as a fluorophore. So fluorophore is nothing but fluorophore are the compound which perform the fluorescence phenomena, and fluorescence phenomena is whatever she told. So fluorescence phenomena. Now what is the fluorescence phenomena? So first we need to understand this. Is this up to here is clear? Okay, अब देखिए fluorescence phenomena. जैसे when we started the class, यहाँ से निकल के हम ऊपर गए, तो ऊपर चले गए आप, फिर नीचे आए, फिर आप बड़बड़ाते हुए कुछ इधर खेलते हुए कुछ उधर खेलते हुए. सो सेम वे, अब हमने कहा आपसे कि आप ऊपर जाओ, तो आप चले गए, फिर आप लौट के आ गए. सो सेम वे, दिस फ्लोरोफोर मॉलिक्यूल, देयर इज सम excitement on the electron. So electron get energized. So it will leave its orbit. Means it will be like the electron is in resting state. ऐसे अर्थ में electron ना बात करें बोलता. Electron is a resting stage. So when you excite with some light, you gave some energy to it. So now it will get excited and it will go into the higher energy state. Okay. Now in the higher energy state, same as कोई भी हमेशा ऊंचाई पे नहीं रह सकता, same way electron भी नहीं रह सकता. Now anyhow it has to come on its जो original state है, it has to return. So similarly, when it get excited and reach at higher energy level, and then after few seconds it will back to its ground state. That's a better terminology. Ground state, higher energy. हम लोग मिट्टी के साथ ही और yeah, these are the terminology that you can use. So when it returns the lower energy state, it will release energy. Okay, and it will, and definitely when it is releasing, it will be lesser energy. जब आप नीचे आएंगे आपने एनर्जी बहुत लेस यूज की होगी, चढ़ने में ज्यादा एनर्जी. Okay, so definitely जब आप ऊपर गए, you lose some energy, and now when you get back, you will release some energy, and accordingly there is a change of the wavelength, the emission of the light. That light, the light which is emitted, the wavelength got changed. Okay, now this is a little bit complicated. Let me explain to you. So, is there anything? Chocolate? आपने एक फॉर्मूला E is equal to S by lambda. So this is you can write E is equal to HT by lambda. So what the important, the focus point which we are going to understand here, E is inversely proportional to the lambda. So if lesser energy, it means more wavelength, and if there is a higher energy, then it is less wavelength. So the excitation light is lesser wavelength, and the emitted light, so excited light means electron was at its ground level, you do excite it so that it go to their higher energy level. So then you need to excite with the lower wavelength light. Okay, and then it will emit the higher wavelength. So there is a shift of wavelength. Okay, and this we know as a Stokes shift, and that we are going to understand. But you understand the fluorescence phenomenon. Okay, and here you just understand the fluorescence phenomenon and what exactly the means. It's a little bit complicated for the biology student, but I tried my best to explain you in a later terminology that when there is an incidence of light, and this is known as excitation, because something is getting excited. जैसे मैं आपको एक्साइट कर दूं, मैं आपको बहुत ज्ञान की बातें दे दूं, आप एक्साइट हो गए घर पे जाके आप पढ़े करके 2-3 दिन बाद फिर सब हो गए और फिर से अपने ग्राउंड स्टेट पे आ गए वापस. So same way, the जो always any compound or any material cannot be it's always in excited state. It has to come down. The same way, electron is also the light which is excite the electron is an excitation, and then when it's returning at its lower or ground level, it will emit the light that is known as a emission light. So there are few terminologies which you need to understand to better understand this concept: excitation light, emission light. Okay, and this is the very beautiful picture which is you can see here. You saw cell here. It's color. No color is there, but here you saw that beautiful color, that nucleus is stained in a blue, and this is cytoskeleton. जैसे हम लोगों के पास बोन होती है, कभी हम खड़े अगर बोन हमारे पास ना हो तो इट्स वेरी हार्ड फॉर अस. सेम वे सेल हैज अ ह्यूज साइटोस्केलेटन. आपने पढ़ा होगा एक्टिन फिलामेंट, मायोसिन फिलामेंट, these are the filament. सेल का भी अपना एक स्ट्रक्चर होता है. ऐसा नहीं कि राउंड इसलिए कि बबल जैसा है कि गुब्बारे जैसा फूला हुआ है. No, it has the various protein which help to provide shape to these cells. Okay, got this point? So here in this we are trying to visualize some cytoskeleton protein along with the nucleus. This is the nucleus and then this is the cytoplasm. You clearly understand. Whenever we observe that where is the nucleus, nucleus is center point. Now rest of the thing is cytoplasm, and in cytoplasm you can see this is a baby-like structure which is cytoskeleton protein. Everything inside this cell is protein. Archie, anything is protein. So it's very hard to understand to see the cytoskeleton protein in this cell. In the phase contrast microscopy, you cannot observe any protein or any organelle or any nucleus. जैसे समझ जाते हैं कि ये dots जो है वो nucleus होते हैं. It's just prediction. But then you put some color, then you come to know if your dye, like here it's violet color, it's nucleus system. Okay, you got this point? So why we do fluorescence microscopy, or why we perform fluorescence microscopy, or why we perform the fluorescent staining to visualize different protein component of cell. It will give you better visualization. So there is like if anyone can tell us, like here we saw this protein. Okay, so here you saw the cytoskeleton protein. Any other method you know that by which we can measure the protein? Chromatography. Okay. Anything? Western blotting. Western blotting is also same way. और उससे क्या प्रोटीन डिटेक्ट करता है. बट वहाँ पे क्या होता है कि आप नायलॉन फिल्टर में मतलब मेम्ब्रेन होती है, उसमें आप प्रोटीन को सेल से निकालते हैं, फिर डाउन उससे फिर. बट हियर you can, if cytoskeleton protein is more, you will see more green. If cytoskeleton protein is less, you will see the less. So it's a direct evidence. Again, Western blotting is not a direct evidence. And this fluorescence microscope is providing a direct evidence which is more acceptable in publication. अगर आप Western blotting भी करेंगे तो कोई गलत नहीं कहेगा. But this is here you are producing image. Image is always good for making anything or making any conclusion to convince anyone. Okay, got this point? So now you understand that I always prefer, wherever I go, I always looking for the fluorescent microscope. This is my, like, the first thing which I can think of is because I worked in the cell biology area. So whenever I want to estimate or I want to look for any protein, I always think of fluorescent microscope. This is the first thing which I always order. मणिपाल में गया तो वहाँ भी मैंने fluorescent microscope order कर दिया, पर तब तक यहाँ हो गया जब तक यूज किया तब तक इधर आ गया. तो unfortunately वहाँ से लेने पे यहाँ पे आया, फिर मैंने सोचना शुरू किया. Now we should purchase of fluorescent microscope because the idea is again to give the direct evidence. Whenever we do any publication, we can provide direct evidence. Okay? Is that okay to all of you? Now let's understand a little bit about the history. So again, because the session is getting recorded, so I was not thinking that this is going to be recorded. So this I downloaded from the internet. It's not my own, but it is again it's used for the teaching purposes. So if you are downloading any content from the internet, from my knowledge, best of my knowledge, I understand that's fair. Nothing, no conflict with it. So again, this I downloaded from the internet, and so Sir George G. Stokes. So whatever the Stokes is that we are talking about. So on his reputation, or to give the credit for this fluorescence, that's why there is the wavelength shift in the emission light and excitation light is known as a Stokes shift. Okay, so because of this scientist. Okay, so Sir George G. Stokes. So this phenomena, fluorescence, was known by the middle of the 19th century British scientist Sir George G. Stokes first made the observation that mineral fluorspar exhibit fluorescence when illuminated with the ultraviolet light, and he coined the term fluorescence. So he saw that when UV light is fall on something, the mineral fluorospar, there is a fluorescence, means it will give some color. Okay, so that observation he made first time, and he also coined the term fluorescence, and that's why means you should know about this scientist, the contribution of this scientist. So that's why we should know name of this scientist. Okay, is this okay? Is this clear? This is little bit about the history of the fluorescence microscopy. So now there is a difference between what is the difference between conventional and fluorescence microscopy. The conventional microscope uses the visible light, which is range about 400 to 700 nanometer, to illuminate and produce a magnified image of a sample. So what it will do? It will just like जब किसी आप डार्क एरिया में जाते हैं, जब आप कभी मान लीजिए पार्क में गए जहाँ लाइट बिल्कुल नहीं है, तो आप क्या करते हैं? लाइट जला, यू इल्यूमिनेट। नाइट टाइम आप ड्राइविंग कर रहे हैं, तो आप कभी ऐसा नहीं होगा कि बिना हेडलाइट जलाए और न चलाते चले जाए, पता चला आपको तो कोई नुकसान नहीं होगा, लेकिन दूसरा आदमी आपको हिट कर देगा। सो because you want to see something. Similarly, the conventional microscope use visible light to just illuminate that. That is the nothing else. No another thing is going on there. Okay, so produce a magnified image of a sample. Now a fluorescence microscope is a little bit uses much higher intensity source. So as I told that it needs excitation. So energy will be high, not lambda. Okay. So definitely if energy is high, lambda will be low. So uses a much higher intensity light source which excite a fluorescent species. Means fluorescent species means the species which contain the fluorescent dye. Again, ऐसा नहीं होता कि नेचर में मैंने आपको बताया कि सेल में ऐसा नहीं फ्लोरोसेंट मॉलिक्यूल पड़ा हुआ है, उसको इमिट किया और फिर प्रोड्यूस किया। एक एग्जांपल आप बता सकते हैं कि जहाँ पे फ्लोरोसेंस फिनोमेना नेचर में देखने को मिलता है, firefly, and what it contains? Lucifers. So that's like but that's a different. Again, that is also depend upon the energy. Firefly जब बैठी रहती है तब कभी नहीं दिखता, जब वो उड़ती है तभी जब when it fly, only then you will see the color. But when it sits, you will not. So because when it fly, it produces more energy. And that energy will create the luminescence. Okay, so that's a separate thing. But in nature, the firefly is one of the examples. Then you can understand, you can track. Yes, the firefly is going. So when excite a fluorescent species in a sample of interest, this fluorescent species in turn emits a lower energy light of a longer wavelength. Now don't get confused. It's mixing. So when there is a high energy, there will be lower wavelength. That produces magnified image instead of the original light source. Okay, so you got this point. So here there is a excitation light and emission light. So there is some light which is exciting, is not same is emitting, but it's a different wavelength light is emitting. So that's what the fluorescence phenomenon. Okay, got this point? Is that clear? So this is the difference between conventional and okay. So this is the image, and this is not very important. I will share this slide. You can read it. So because fluorescence is called as a poor light because it does not come from the hot source, or because it is not coming from the hot source, but it is a high energy light. Okay, high energy light. So that's why it is also called as a cold light. And here in the picture you saw that this round color is a fluorescent emission. Now, again, this is also self-explanatory. Whatever I already told you about fluorescence microscopy. So, fluorescence microscopy is a unique way of using microscope to discover a fact about the specimen that are not shown by the standard bright field microscopy. So, whatever you cannot quantitate or observe by the conventional microscope, that you can observe from fluorescent microscopy. In bright field microscopy, specimens are illuminated from outside, below or above, and dark objects are seen against the light background. In fluorescence microscopy, specimens are self-illuminated because you added the fluorophore. आपने उसमें fluorophore you added, so that's why it will be illuminated, self-illuminated. Self-illuminated by internal light, so bright objects are seen vivid color against dark background. Bright objects against dark background are more easily seen, and the characteristics of fluorescent microscopy make it very sensitive and specific because it produces different color, so you can observe different things in a much easier way. You can, on the same time, you can observe nucleus, on the same time you can observe cytoskeleton, on the same time you can observe the, let's say, any other organelle in an effective manner, because let's say you stain with a different color. You stain nucleus with a violet stain. So that's what the first picture you can see here. All different things are easily observed. So this one is nucleus, and this cytoskeleton, this is something else, maybe actin or myosin stain. So everything can be observed very effectively. Okay, so this is the advantage of the fluorescent microscope. So, again, to understand how the fluorescence microscope works. Okay, so this is a energy source which is giving the energy to the this specimen. Okay, so this is one light source, or what you call this excitatory light. Okay, so this will create excitation here. So this gives the color, and then there is, this is known as an excitation filter. Okay, so there is no naming, but this is excitation filter, and this is the emission filter. So again, what is the meaning of the filter? Filter, जैसे filter use करते हैं, filter साफ पानी के लिए आप कौन सा filter एक्वागार्ड यूज करते हैं, या फिर जिसका नॉर्मल स्ट्रक्चर रहती है वो एक्वागार्ड वही करती है, ऐड आरओ फिल्टर, जो मतलब इसे it's filter something, it will हाँ, means only pure water you will get, उसमें और component मिला वो भी अपना नहीं है, nothing like that. Same way, these filters will help to pass on only excitation light, not any other light. Means, see, in case of visible light, there is a big pure, you understand, violet, indigo, blue, red. There is a visible light contains various spectrum of the light. So whatever the excitation light is, जो लाइट एब्जॉर्ब, जिस वेवलेंथ की लाइट उस फ्लोरोफोर मॉलिक्यूल को एक्साइट करेगी, this excitation filter will pass on to only that light. Here in this cartoon you can see that only violet is going. सारे कलर are, but only violet is going. So it might be the point is that maybe the violet color means it's just representation. Only the violet color is giving excitation to this fluorophore molecule. ऐसा नहीं कि वो cell को excite कर देगा. Nothing like that. It will excite the fluorophore molecule because fluorescence phenomena will occur only to the fluorophore molecule, because your specimen contains this fluorophore dye. We don't have the fluorophore dye inside our body. Okay, so in the specimen you need to add on the fluorophore molecule, and here you can see from the excitation filter, only the excitation light is emitting on this fluorophore molecule. As the excitation is going on, then definitely there will be emission. So this will take the energy, the electrons. ऐसा नहीं कि ये मॉलिक्यूल अब ऊपर चला जाएगा, नहीं, ऐसा नहीं, लाइट जाएगी. Okay, so this will, the electrons in this fluorophore molecule will go to the higher energy state, and after few seconds, very quick, it's a very quick, 10 to the power minus 4 seconds. It's come down the lower state, and then it will emit a light, and this green light is a representation of the what light? Excitation. Excitation. Excitation light. Excitation light is clear. मैंने आपको दो ही चीजें बताई, चीज खास कर मैंने आपको बताया था कि आप जो लाइट डालोगे, तो मैं आपको अब एक्साइट किया, एक्साइटेशन लाइट, अब फिर आप इमिट करो, you will emit, so that when it will get on the lower energy state, it will emit some light. So wavelengths are different, that's why their naming is different. When something is emitting is just a name hai. Excitation means only for excitation. Emission means something which is emitted from that molecule. So this fluorophore molecule will emit some light. So that is known as the emission light. EMISSON, emission light, excitation light. Okay, so now it is clear. So nothing else. Excitation light. Now it will emit some light, and this emission light will be, you are going to observe that light. You got this point? And this will only go to the ocular, means from your eye, where the lens, ocular lens means, you know, the eye lens, where you observe. So only emission light will go. So here the violet light comes, but the emitting light is green light. Okay, so this is emitted, and then what filter it is? इस फिल्टर का क्या नाम हो सकता है? एक कॉमन सेंस क्या हो सकता है? उसका एक्साइटेशन फिल्टर, इसका एमिशन फिल्टर, ये तो कोई भी बता सकता है, नहीं जिसको फ्लोरेसेंस माइक्रोस्कोपी पढ़ा वो भी बता सकता है, ठीक है? Now and then this is another thing which is known as a dichromatic mirror, because this mirror, two different type of light. Dichrome means color, di means two. Okay, so dichromatic mirror. This ये mirror जैसे नहीं, कभी कभी आप देखते हो यहाँ mirror लगा तो वहां की चीज देखते हैं. So this is known as a dichromatic mirror, because it will pass all the light. अगर ये dichromatic mirror नहीं लगा होता तो ये लाइट इधर जल जाती है, कहीं उधर जाके और दिन पृथ्वी में मिल जाती है, ऐसे ये ऑब्जेक्ट पे आते हैं. So this dichromatic mirror, यहाँ पे इसका कोई ऐसा चीज नहीं है, but two different type of light is falling on this mirror. That's why this mirror is known as dichromatic. Science is like, you know, science always make a sense. साइंस में हमेशा सेंस रहेगा, ऐसा नहीं कि कुछ भी नाम रख दिया इसका. हाँ, हम लोगों की तरह कि जैसे हमारा एक स्टूडेंट था, उसका नाम था तेज, तो बिल्कुल भी तेज नहीं था. सो यू नो, सो यहाँ साइंस में फर्स्ट यू नीड टू ऑब्जर्व द नेम. इफ यू ऑब्जर्व द नेम यू विल अंडरस्टैंड व्हाट एग्जैक्टली इट इज फॉर. तो इसीलिए फ्लोरेसेंट माइक्रोस्कोपी, पहले आपको हमेशा साइंस में हमेशा याद रखिए क्यों उसका ये नाम पड़ा. किसी पढ़े लिखे स्ट्रोक साहब ने नाम रखा तो कुछ सोचा होगा उन्होंने. So that's why you should first don't means, yeah, definitely there is some point that you need to memorize the thing, but sometimes you can make a sense that why it is known as a dichromatic mirror. Okay, so this is the very general outline for fluorescent microscope, how the fluorescent microscope works. Okay, so this is excitation filter, this is emission filter, this is ocular, means from the lens where you are viewing the things, and this is a dichromatic mirror, and here is the objective lens piece. This objective lens will help us to object only to the specimen, so that your light should go only specimen, not anything else. Okay, so this is the simple way how a fluorescent means. Definitely, अगर आप fluorescent microscope कभी देखेंगे, fluorescent microscope now this is this ऐसा नहीं कि वो मैंने कार्टून में उसी की तरह दिख रहा है. No, this is the, but again you can make sense where this is the light source, this is the ocular lens, might be there some dichromatic mirror there, and there are some filters as well, excitation filter, emission filter, and then this is the place, this is the objective lens, and this is the place where you put your source. Okay, so this is that's all for today. Any question from your side?
Yes, sir.
Yes.
जैसे आपने बताया कि सर ब्लू लाइट साइड में हमें जो शो हुई एट द ग्रीन लाइट, तो क्या सर कंडीशन अप्लाई है कि जब ब्लू एक्साइट होगी तभी हमें ग्रीन दिखाई पड़ेगी, अगर वो दूसरी लाइट हो तो क्या हमें ग्रीन दिखाई नहीं पड़ेगी?
बिल्कुल सही. Means whatever she, yes, it's defined. It's defined that I will teach only to the fluorescent microscope. Not anything I will going to teach the let's say any microbiology thing. No, whatever, because this is defined that this light, when excitation light will be the violet and emission light will be green. This is defined, and in the next class I will show you some slide. One slide means this. I was thinking that I will show you. Otherwise, there will be some dye list where you can see that. So here today is not in this slide, but I will show you that yes, this is specified that this light, some dye will produce green color, some dye will produce only red color, some dye will produce any other color. So this is defined. This is the beauty of this fluorescent microscopy. In that way, on the same time, if something is red with this, let's say you took a fluorophore molecule which is specific to the cytoskeleton. Okay, so then if you saw the green light, so you saw that, you understand that okay, cytoskeleton protein is there. Let's say you stain lysosome with the red dye. If there is a lysosome, you can see that there is a whatever the organelle which is red in color, it must be lysosome. So by this we can we perform different experiment and we draw various conclusions. So in next class I will continue with this one and also give you some fluorophore molecules. So there are various types of fluorophore molecules, but one thing is very common in all fluorophore molecules, that excitation light lambda should be less, emission will be lambda. This is very common in all the fluorophore, whether they are producing any type of color. So if you view according to the wavelength, the excitation wavelength will be always lesser and emission wavelength should be always, if you get a chance after reading this one, if you will see some fluorophore molecule and you will see the excitation and emission filter, you will find that lambda is, excitation is low, emission is high. Okay, so it is specific. Yeah. This is Stokes law. So there is a shift in wavelength. That was the लोगों को नहीं विश्वास है, एक चीज गिर रही दूसरी चीज कैसे निकलेगी. तो so then it's a, yeah, this is the Stokes shift that we call this Stokes shift, and this is change in wavelength. Anything, but you now understand that because why it is called Stokes, why not it is called Rajeev will shift or something, any other shift. So because of that, he coined the term the fluorescence. In that honor, it was kept as a spokesman. Okay. Anything else? So yeah, that's all for today, and we will continue with the fluorescence microscope. And yes.
Sir, in fluorescence microscopy, we can visualize only dead cells.
No, live cells.
Live cells.
Yes, she can visualize, but हमको उनको मारना पड़ेगा.
Both dead and living cells.
That I will tell you means yes, definitely you have to stay when they are live, but for visualization आपको इस microscope के नीचे लाना पड़ेगा और live cell को कभी भी अगर आप live cell को ले आए अच्छी बात है ले आए पर आप फिर से जब वापस उसको कल्चर में ले जाएंगे तो वो डेड मतलब कंटामिनेशन हो जाएगा या कुछ it's not because you need to prepare some slide, so all these kept in a slide, so that's what that's why these staining is done in a live cell but observation is in dead cell. This is when फिर अपना वो slide बना के रखेंगे हमेशा के लिए कोई भी आए तो उसको दिखा देगा. But there is one thing, this fluorophore dye is also quenching, so that in term photo bleaching we will read in next class. So anything else? Okay. So that's all for today. And any किसी का आप में से आपका WhatsApp नंबर मुझे मिल जाता so that we can connect. तो उस ग्रुप पे हमें ऐड ना करिए, हमें कोई एक CR बन जाए जिससे ऐसा रहता है. It's all.
This group में सारे टीचर्स ऐड हैं. तो भी नोट्स भेजते हैं उसी ग्रुप में सारे टीचर्स भेजते हैं.
ओके. तो मैं एक अलग ग्रुप बनूँ. फिर ये रहता है उसी से. ऐसे बहुत से मैसेज आते हैं. हाँ, yes, I will give you my. हम इसको बंद कैसे करें?